In this video, you will learn how action potentials are generated and propagated. Then you will learn about the structure and function of a synapse. Finally, we will see how these topics come together as you learn to compare and contrast graded potentials and action potentials. Information is rapidly carried through neurons in the form of electrical nerve impulses called action potentials. In this example, sensory information about the temperature of the water is carried to the central nervous system in the form of action potentials that travel along the axons of afferent neurons, and motor commands are carried out to the muscles in the form of action potentials that travel down the axons of efferent neurons. Neurons in the spinal cord and brain also use action potentials to carry information as they integrate the sensory input and generate the output motor commands. Action potentials are electrical signals that result from ions rushing in and out of neurons. When the sensory receptors in our skin detect the temperature of the water, ion channels open, allowing sodium to enter the cell. The sodium ions carry a positive electric charge across the cell membrane. To understand the fascinating topic of how neurons carry information, we will need to zoom in and think about how the electrically charged sodium and potassium ions contribute to the separation of charge across a cell membrane. All excitable cells, including neurons, have a membrane potential, a separation of charge across the plasma membrane that can be measured in volts. The value of the membrane potential can change when a neuron is receiving or transmitting information. At rest, neurons maintain a resting potential of about -70 millivolts. This membrane potential results from unequal ion distribution and permeability. There is more sodium outside the cell and more potassium inside. The sodium-potassium pump creates these concentration gradients. This protein uses ATP to fuel the movement of sodium out of the cell and potassium into the cell. The membrane has leak channels that allow these ions to cross the plasma membrane when the cell is at rest. There are more potassium leak channels than sodium leak channels. The equilibrium potential for an ion is the value of the membrane potential that would produce an electrical force that is equal and opposite to the force of diffusion. The value of the equilibrium potential can be calculated with the Nernst equation. This equation uses the concentration gradient for an ion to calculate the value of the equilibrium potential. While the concentration of sodium in the extracellular fluid is 145 millimoles per liter, the intracellular fluid contains 12 millimoles per liter. Using these numbers in the Nernst equation, we can calculate that the equilibrium potential for sodium is about 66 millivolts. For potassium, the value of the equilibrium potential is about -97 millivolts. This value is negative because the potassium ions have a positive charge, and there is a higher concentration of potassium inside the cell. The Goldman-Hodgkin-Katz equation uses the relative permeability and concentration gradients of ions to calculate the value of the membrane potential. The resting potential of -70 millivolts is closer to the equilibrium potential for potassium than sodium, because the relative permeability of the membrane is greater for potassium. The action potential is a brief reversal of the membrane potential that travels down the entire length of the axon from the axon hillock to the axon terminals. Depolarization, repolarization, and hyperpolarization are changes in the membrane potential that occur during the action potential. Depolarization is when the membrane potential increases, becoming less negative than the resting membrane potential. Repolarization is when the membrane potential returns to the resting potential. Hyperpolarization is when the membrane potential becomes more negative than the resting potential. When a neuron is stimulated, sodium channels open, increasing the permeability of the membrane to sodium. Depolarization occurs as sodium rushes into the cell. If depolarization reaches a critical point, the threshold potential at -55 millivolts, voltage-gated sodium channels open, triggering an action potential. Depolarization occurs during the rising phase of the action potential as sodium enters the neuron. During the falling phase, voltage-gated potassium channels open and potassium exits the neuron, causing repolarization and hyperpolarization. Hyperpolarization occurs at the end of the falling phase because more potassium channels are open compared to when the neuron was at rest. Action potentials are all-or-nothing events that are generated at the axon hillock and travel the full length of the axon without losing strength. As sodium enters the neuron, it spreads down the axon, stimulating voltage-gated sodium channels that open, producing more depolarization in a positive feedback loop. When an action potential arrives at the axon terminals, depolarization will stimulate the release of neurotransmitters from the axon terminals into the synapse. These neurotransmitters are chemical messages that will send information to the next cell. The synapse is a junction between two cells where information travels from one cell called the presynaptic cell to another cell called the postsynaptic cell. Calcium ions are involved in stimulating the release of neurotransmitters from the axon terminals of the presynaptic cell. When the action potential arrives at the axon terminals, depolarization stimulates opening of voltage-gated calcium channels. Calcium rushes into the axon terminals and stimulates exocytosis to release neurotransmitters from synaptic vesicles. Neurotransmitters diffuse across a small space called the synaptic cleft and then bind to receptors on the postsynaptic cell. After binding to the receptors, neurotransmitters have to be removed from the synaptic cleft to terminate the signal. Neurotransmitters can be removed by reuptake when they are transported back into the presynaptic neuron. Neurotransmitters could also be broken down by an enzyme, or they could simply diffuse out of the synaptic cleft. Graded potentials are localized changes in membrane potential that occur when a neuron is receiving information. They are short-lived and decrease in strength as they spread. Graded potentials often occur when neurotransmitters bind to receptors on the dendrites of a postsynaptic cell. If the neurotransmitter stimulates depolarization, it produces a type of graded potential called an excitatory postsynaptic potential. This type of neurotransmitter is excitatory because depolarization brings the membrane potential closer to the threshold for an action potential. One excitatory postsynaptic potential may not be strong enough to reach threshold. Summation is when multiple graded potentials add together. Summation of excitatory postsynaptic potentials from multiple inputs can add up to reach threshold at the axon hillock. If the neurotransmitter is inhibitory, it causes hyperpolarization in the form of a graded potential we call an inhibitory postsynaptic potential. The sodium ion channels that open to produce excitatory postsynaptic potentials are receptors for neurotransmitters called ligand-gated ion channels. The neurotransmitter is the ligand that binds to the receptor and stimulates opening of the channel. Mechanically gated ion channels are found in sensory receptors. These ion channels produce graded potentials when they open in response to a mechanical stimulus such as pressure or vibration. In our example of a sensory receptor detecting the temperature of water, sodium enters the cell producing a graded potential. This graded potential in the dendrites of the sensory receptor is the initial depolarization that initiates the action potential that travels up the axon. In contrast to graded potentials, action potentials are all or nothing and travel without diminishing. A stronger stimulus might increase the frequency of action potentials, but the size of the action potentials cannot change. In summary, neurons transmit information by changes in membrane potential. Graded potentials occur locally and may or may not reach threshold. If threshold is reached, an action potential is triggered and propagates down the axon. Neurotransmitters carry the signal across the synapse, continuing the chain of communication.